and
physical slides. Yes.
Yeah, exactly. Yeah.
Well, Walter, I think that we are ready to go now.
Sure.
We are because we are around 40 students.
Mm-hmm. Good.
So I'm gonna give you permission to.
Yeah.
I'm gonna give you superpowers, then you can.
Give me superpowers. Excellent.
Yes, indeed. Okay. Now you can.
Good. Can share the screen?
Yes, yes, indeed.
Okay, I'll do that. And so you see my screen now and my
your pointer? Yes.
My pointer. Very good.
Mm-hmm.
So is it okay? I'll talk about an hour.
Yeah, it is.
I prepared for about an hour, and of course, you know, if anybody wants to interrupt me, I'm probably going to make a little break after each of the sections, and then maybe just quickly ask if there's a question, but then we can also discuss at the end.
Mm-hmm. Okay.
So I'll be. Thank you, Louise, for the invitation.
Yes.
I'm going to be talking about muscles and muscle functions. And specifically, what I would like to do is talk about this particular outline here. I talk first about a little bit about what I call the basics of muscles, which is the structure and the sliding filament and cross-bridge theory. And then I go into what I consider the three major mechanical properties of muscles: the force length, force velocity, and history-dependent properties, and then give a little bit an explanation of those, and then show an example from sport or an applied science where we used these type of relationships and these type of properties to gain more insight into human movement or athletic performance. So I'm going to start out with talking very briefly about the structure of muscle, just to remind everybody what we talk about. So we'll be talking about skeletal muscle, not smooth muscle, not cardiac muscle. So the muscles that you have in your arms and legs. And as you know, they come in a variety of different shapes in terms of the fiber arrangement, where we have muscles with essentially parallel fibers, and here as well, the fusiform muscles. And then muscles where the fiber direction is at a distinct angle to the long axis of the muscle, which we call pennate muscles. And if you have one of those directions, then we call it unipennate. And if you have two directions, we call it bipennate. And if it's more directions, we call it multipennate. And there's very distinct reasons, functional reasons why we have muscles of different shapes and different arrangement, but I'll not be talking about that today. So I want to go directly into the more cellular molecular aspects of muscles. Here we have an entire muscle, and we know that muscles typically, especially bigger muscles, have subdivisions into fascicles, where fascicles are surrounded by connective tissue. And then in these fascicles, we have isolated muscle fibers or muscle cells. And if you then go to an even lower level, where we work a lot, when you then go from a muscle fiber, then usually muscle fibers in skeletal muscles of vertebrates are made up of these myofibrils, which are serially arranged sarcomeres. And you have about 2,000 to 4,000 myofibrils in a single muscle cell or in a single muscle fiber. And very often we work on the myofibrillar level, and sometimes we even work on the isolated sarcomere level. And so here you have a sarcomere. So a myofibril is made up of these serially arranged sarcomeres, about 400 sarcomeres per millimeter of myofibril. And the sarcomere, of course, has these two contractile proteins in the middle, the myosin filament, and then here at the end, going into the Z line, the actin filament. And again, we work a lot on that particular level with isolated filaments as well. However, what is often forgotten is that when we look at the sarcomere, here is the Z band, here and here, and the myosin filament in the middle on green, and the actin filament here in red. What is often forgotten is a third filamentous structure in muscle, and that is titin. And so here we see titin in blue that goes from the middle of the sarcomere and is fixed onto the myosin filament and then runs freely across the I-band region along the actin filament. And then here at the very end inserts and connects with the actin filament, and then together with the actin filament goes and anchors itself into the Z-band. And in this particular region here, we know that you can extend the titin, and here are some molecular structures of titin that are extensible. So for example, these brown rectangles here represent the immunoglobulin domains that are normally in the relaxed state about 3.8 nanometers long. And when you unfold them, they have two unfolding sequences. When you fully unfold them, they get to 28 nanometers. So in other words, each of these immunoglobulin domains can increase its length by about a factor of 7. And then the same is true for the N2A and the PVK regions of titin. And I'll be talking about that a little bit later. So having seen across a sarcomere, I'm just very briefly going to review for most of you about the cross-bridge theory and the sliding filament theory. So how we imagine that muscles contract and produce force. And as you know, since 1953 and 1954, we think that there is this sliding of these filaments going on where the actin filaments slide past the myosin filaments. Many of you will know that before the 1950s, we thought that shortening force production was achieved by a shortening of the myosin filament here. And then Hugh Huxley and Andrew Huxley in these publications in 1953 and 1954 showed very nicely that no, the myosin filament stays at about a constant length and therefore could not be the reason for force production and shortening of muscle. And that's when they came up with the theory that we have these actin filaments sliding into the myosin filaments and bringing the Z lines closer to the center of the sarcomere, thereby shortening sarcomeres and producing force. And this was all first described by Andrew Huxley in 1957. Here is Andrew Huxley with his wife, Regenda, when they visited Calgary. And so what he proposed is that we have a myosin filament shown here schematically in a very engineering-like drawing. And then he assumed that there would be side pieces, which we now say cross bridges, that are attached to an elastic element. And then this cross bridge would move around according to Brownian motion. And then every once in a while would be in the neighborhood of an actin attachment site. So here is actin with a symbolized actin attachment site. And then the myosin would attach to the actin. And through the force of this spring-loaded system, it would then exert the force and pull or tend to pull the actin to the left in this particular case. So this will be with the Z line here. So this will be an actin on the right-hand side of a half sarcomere. On the other hand of the sarcomere, of course, you would have exactly the opposite direction. And then he proposed that this was all governed by a rate constant of detachment, which he called G, and rate constant of attachment, which he called F. So all this is governed by certain rate constants that follow precisely defined laws, and he described that mathematically in 1957. And a more recent version of this looks about like this, where we have the myosin filament here, we have the elastic connector to the cross-bridge head here, and then we have the cross-bridge head here that attaches to the actin filament. And through the hydrolysis of ATP, 1 ATP per cross-bridge cycle, we have this interaction of the cross-bridge with the actin filament. And through a rotational movement, the actin filament is then pulled past the myosin filament. And here we look at the left-hand side of a sarcomere. That's why the actin was pulled from left to right in this particular drawing. So from this, which is really all available in textbooks, we can look at some of the properties of muscles. And the first one that I want to talk about is the force length relationship. And the force length relationship essentially describes the maximum active isometric force of a muscle, shown here on the vertical axis, as a function of length. And the length is given sometimes as the whole muscle tendon unit length or a fiber length. And in this classic presentation by Gordon et al. In 1966 in the Journal of Physiology, it's shown as a function of a sarcomere length. And so here, this is for a frog sartorius muscle. We see that you reach an optimal length or a plateau region where the muscle can exert the maximum force. And for frog, that happens to be at about 2 to 2.2 microns of sarcomere length. And when the muscle gets shorter, it loses force. And when it gets longer, it loses force as well, which we then call the ascending limb and the descending limb of the force length relationship. And it's very well accepted today that the isometric maximum force depends on the length of the muscle and is described in this particular way. This relationship is nicely explained by some elements of the cross-bridge theory, because when we look at the descending limb here at the plateau, we can explain this very nicely with the amount of overlap between the actin and the myosin filament, and therefore the number of cross bridges that can interact between actin and myosin. So here at C, at the long level where you have no force, that indicates a very long muscle where the sarcomeres are so long that the actin doesn't overlap with the filament, with the myosin filament anymore, and therefore there's no interaction of the cross bridges with the actin filament, and therefore there's no active force. If now the muscle and the sarcomere shortens and the Z line comes in here, then you start gaining overlap between actin and myosin, and more cross bridges can interact with actin, and then the force proportionally increases until you reach point B. And at point B, your actin filament is overlapping all the possible cross bridges on the myosin filament. And therefore you get a maximum amount of force because there's a maximum number of cross bridges that can interact with the actin. If you now become even shorter and go to A, then you go from this position to this one, where the actin filament now goes into this empty zone in the middle of the sarcomere, which we call the bare zone. And in this bare zone, you have no increase in force because there's no cross bridges there, because the cross bridges on this side and on this side are 180-degree oppositely directed. And so here you only have the tail portion of the myosin and not the head portion. And that's why you never have any cross bridges in the middle of a sarcomere. So in sport, or when you do ergonomics, work, then of course you would like to work with a muscle at an optimal length or close to where it can exert the maximum force. You don't want to work here. You don't want to work here where you have very little active force available in the normal case. And so we had asked ourselves the question whether or not in athletic movements you would see that athletes work in this particular range of the force length relationship. And one of these studies was on cyclists. So we asked the fairly simple question, do cyclists use muscles in an optimal way according to the force length relationship? And we did a variety of experimental and also a couple of theoretical studies on this. And I'm just going to show you some isolated results. So here, for example, we had 15 cyclists where we measured the torque angle relationship of the knee extensor muscles, and simultaneously with ultrasound measured the lengths of the fascicles of the vastus lateralis to obtain a torque angle relationship and also a relationship between the torque and the fascicle lengths of the vastus lateralis. And then we put these same people on a bicycle ergometer, and they adjusted the seat height until they were comfortable. And then they were cycling, and when they were cycling, we measured the fascicle length of the vastus lateralis with the same setup as we had before. And we were particularly interested in here in this power phase of cycling, what the fascicle length would be. And so here we then got from these 15 people the following quadriceps force as a function of fascicle length relationship. And of course, we have here error bars because, you know, the torque produced and the force produced and the fascicle length was not the same for all of them. So this represents a standard deviation there. Then we normalized this force length relationship. And since we didn't have values here at very, very long muscle lengths, we extrapolated this linearly. And then we checked for cycling at 80 revolutions per minute for a maximal effort, where would the fascicle length be during the cycling activity? And what we found was this: at the beginning of the power phase, the fascicle length on average was here at about 1.25 of the normalized optimal fascicle length. And then it shortened to the end of the power cycle in cycling. And then we were at a length of about 0.9 of the fascicle length. And as you can see, this very nicely kind of captures the peak area of where the muscles are strongest. And so therefore, for this maximal cycling activity at 80 revolutions per minute, we concluded that cyclists appear to use the knee extensor muscles optimally from the point of view of the torque angle relationship. So they work at a good length. And when they adjusted their seat height, all these cyclists were in a position to adjust their seat height in a way that they were working optimally. As you know, if you've ever cycled on a bicycle that's too small for you, for example, and you sit very low, then it becomes very hard for you to cycle. And one of the reasons is because you're really not working at a very good length in terms of the force length or torque angle relationship. Another question we asked ourselves is, what is the optimal direction of crank force application in cycling? Because we know that if the torque at the knee joint is maximal, that's really good for cycling. But we also know when you are in certain positions on the bicycle and you activate the knee extensor muscles, they give very much a tangential component to the crank rather than one that's perpendicular to the crank. And why is that important? It is important because people have argued that ideally you would like to always push the pedal exactly perpendicular to the crank. So if the crank here, let's say, is exactly in a horizontal position, you would like to push directly down. And this, of course, comes from the work definition or the power definition. Power, of course, is the dot product of two vectors, the force vector, which points in this direction, and the velocity vector. And at this point, the pedal's velocity would also be going down and be in the same direction. And we know that the dot product, which gives us the power output, the instantaneous power output, the dot product is also the magnitude of the force times the velocity multiplied by the cosine of alpha. And if these two vectors point in the same direction, then the angle alpha is 0 and the cosine becomes 1, and you have 100% efficiency in terms of work production. In contrast, what people seem to be doing in reality is that in this phase, they don't push in that direction, but they push slightly forward, which then means only a component of this total force would contribute to the work production, and the other component that's directly along the crank, of course, would not produce any mechanical work, and therefore has been considered as being wasted. And people have quantified an efficiency of cycling. And the bigger this component is, the horizontal one relative to the vertical one, the bigger, the less efficient people have been said to be cycling. And there have been investigations in trying to train people to really be more efficient in their cycling. However, as we know from many, many studies, I'm just showing one very early on, people don't do that. So here, in the ideal case, mechanically speaking, if the crank is up here, you should push in the direction of this white line, but on average, people push in a direction that's quite opposite to that. And similarly here and here, the white line and the yellow line, the white line being the one that you would expect them to push, and the yellow one where what people usually do, are very, very different. And not only recreational cyclists, but also world-class cyclists do that. And then the question becomes, why would they do that? Because from a mechanical, purely from an engineering point of view, it's quite clear that you would like to always push perpendicular to the crank and not at some angle. And we hypothesized then that maybe applying force perpendicular to the crank might not necessarily be optimal from a biomechanical point of view, although from a mechanical point of view, you could argue it is. And why would that be? So we took 35 subjects and had them push against the crank at 5 different angles in the power phase. And we did that with a maximum effort in what we call an unconstrained situation and a constrained situation, which I'll explain in a second. And then we also measured the corresponding electromyographical activity and also modeled this with a theoretical model. But what do we mean by unconstrained and constrained? What we mean by that is when we asked the people to push the pedal in an unconstrained manner, they did not have to worry about what the direction of the resultant pedal force was. And when we asked them to push in a constrained situation, then we asked them always to push exactly perpendicular to the crank for each of the different crank angles. And we gave them feedback, and they had to be within 5 degrees plus or minus of the direction that is perpendicular to the crank. So we did that, and here is just one example. This is the mean from these 35 people that we did that with. So on average, the 35 people pushed in this particular direction with a force of about 500 newtons. This is a 200 Newton thing. And so the tangential component then would be this one. So this is the component that produces work. And then this component here that's exactly parallel to the crank would not produce work. So this is the unconstrained force. And this is the component that produces work or power in cycling. And now we ask the people to push exactly in that direction. And when we asked them to do that, then the force was only about half as big. Or in other words, they were much weaker when they were constrained to push in this direction than they were when they could push in any direction. And in fact, the tangential component was about twice as big than when they were asked to push in this particular direction. And this was true for all the angles. If you compare here the white, which is the tangential component of the unconstrained situation, it's always much bigger than the brown one. So here is the brown versus white, brown versus white, brown versus white, and brown versus white. And so in other words, when the people, these cyclists, pushed unconstrained, then it was very easy for them to obtain a component that was equally big as the maximum force that they could exert if they were asked to push tangential to the crank. So we then went a little bit further and we said, well, how much is it actually influenced how the people push in terms of directionality? And so we brought people back and we had them push in a variety of different angles. And as you can see, you then get a force envelope. And it's quite clear that people are very strong when they push in some directions, and they're relatively weak, for example, here perpendicular when they push in certain other directions. And then you can find which of these directions gives you the maximum tangential component. And very often it's not the maximum force that you can exert, but because here the tangential component now is only this long because the angle is very shallow, whereas here, which is a direction that people prefer, here the tangential component is actually bigger. So here, although the force is lower than here, the tangential component becomes bigger. and the tangential component is much bigger than when you push tangentially. So there's an optimal direction to pushing that maximizes your ability to produce force and your ability to produce a tangential component. And interestingly enough, what we found from our cyclists, they seem to be pushing always very close to the direction that allows them to get a maximal force tangential to the crank. And this maximal force tangential to the crank is not achieved by producing force perpendicular to the crank, but is typically achieved by letting people do what they naturally do and push in the natural direction. And as some of you might know, there is coaches and athletes and scientists that advocate very strongly that cyclists should be producing force perpendicular to the crank and try to train them. And I personally think that's really a disadvantage, because when you do that, then although you take care of the mechanics in an engineering sense, you don't account for the biomechanics of the system. You don't account for the fact that the force that you can exert in certain positions when you're on the bicycle depends very, very much on the direction in which you're pushing. So the points to remember here for this first section of my talk were that the force-length relationship can be directly derived from the crossbreed theory. It's directly an associate of the crossbreed theory. It's important to consider for sports where the working range can be selected. I gave the example of cycling, but you can also think about rowing. You can think about certain weightlifting activities. You can also think about starts, for example, in track and field sprint starting, the position that you adopt when you go into the starting blocks, and many other situations where you can maximize the force length properties that you have. And one thing that I did not talk about today, there's not enough time for that, is that we know that force length properties also adapt to the chronic and sport-specific demands. So we have shown in earlier studies, for example, that the force length properties of cyclists and speed skaters are very, very different from those of runners. And you can directly associate those with the functional demands of the sport and the functional demands that are imposed on these muscles during the training activity. So I'm going now to the force-velocity relationship, but if somebody had a very urgent question, then we can quickly stop for that. If not, then I just go on. So let's talk about the second, what I consider the second basic property of skeletal muscle, which is the force and or power velocity relationship, made famous by a publication by A.V. Hill in 1938. And the relationship essentially says that if you have an isometric force, you can exert a lot of force, and your force, shown here on the vertical axis, decreases with faster shortening velocities. And at one point, your shortening velocity will be so big that you cannot really produce any external force anymore. You can then derive from that the instantaneous power output of a muscle by assuming that we calculate power of a muscle as just a scalar product, or I should say just the product of the force magnitude times the velocity magnitude. It's really interesting because for many situations, you could argue that this equation is not quite correct and that the force of the muscle is not going in the same direction as the endpoint of the muscle. But this has just been taken by people, and it's definitely true for isolated muscle and isolated fibers. It's definitely not correct to do that for in vivo human skeletal muscle, although this is typically being done. But what you have here is when you do that, you can take any point on the force velocity relationship, and this one, and then the next one, and the next one, multiply them by one another, and then you get this curve that is now the power as a function of the velocity of shortening. And here power would be zero because force has become zero because the velocity is so fast. And here we have power is zero because we have an isometric contraction and the velocity of shortening is zero. And somewhere in the middle here at about 30 to 35% of the maximum velocity of shortening is where in an isolated muscle or fiber preparation, you get the maximum power output. So is this relatable to the cross-bridge model? Yes, it is, and no, it isn't. The cross-bridge model, as shown here by Huxley, predicts the force-velocity relationship very well, but it doesn't do this because of some structural component or inherent property of the cross-bridges, but it does it because of the way the cross-bridge rate functions of attachment and detachment have been formulated. Andrew Huxley, when deriving the cross-bridge theory, was very careful that he could fit his model to the existing knowledge on the force-velocity relationship, and that could be done by appropriate choice of rate constant, but it does not come out of some structural or other functional component of muscle that we are aware of. So it's something to know. If we now go to the optimal muscle parameter, then of course in many sports, we would like to work, if this is our power-velocity relationship, we would like to work for many explosive type of actions. For example, like shot putting or high jumping or sprinting, you would like to work in an area of the force and power-velocity relationship where your power output is maximized. And again, we have looked at functional power output in different athletic movements. And one that I'm going to show you here as an example is cross-country skiing, and particularly the action of double poling in cross-country skiing. And I realized that in Brazil, not that many people are familiar with cross-country skiing, but I would like to announce that at the last World Championships, which just finished about 2 weeks ago, full four-person team for the men and women and participated at the Nordic cross-country skiing World Championships. So when you look at double poling, we can look at force output at different speeds, and we were wondering if this somehow obeyed the functional force-velocity relationship of an isolated muscle. So since most of you are probably not familiar how double poling looks like, I'm just going to show here a little video where the person here does a double poling action, and you pay particular attention of the arm and pole actions, because the entire propulsion really is delivered to the snow by the poles, and the skis are essentially frictionless and do not allow to produce propulsive force to any considerable extent along the direction of propulsion. So here we go. So here we have the skier going. You see the poling action that he has. And I'm just going to show that once more. And pay attention, all the power in this particular stroke, which is used, this is the main stroke that people use in classic skiing, and is also the main stroke. Double poling is also the main stroke in the so-called skating technique. So it's something that is used all the time and essentially continuously by cross-country skier. So we put people on a treadmill and we had them perform this double poling action at a variety of different speeds while skiing on a treadmill. And that looked something like this. And we had them fully instrumented because we measured the kinematics and the pole forces and the ski forces and the speed of the treadmill and so on, measured all these type of things. And then in this particular case, we're interested in what is the force, which we really calculated as an impulse. So we took the force over the entire contact phase. So we calculate the impulse as a function of speed of skiing from 6 kilometers to 42 kilometers per hour. And you see for these two different techniques, we get a similar curve that is slightly bent, very similar to what we would expect from the force-velocity relationship of an isolated muscle. if we now convert that into a power velocity relationship for the one scaling action, we see power here as a function of speed. And you see that again, we get a peak power output, instantaneous power output, very similar to what we see when we look at an isolated muscle. So here is the maximum for this particular skiers. And normally skiers in a classic race, when they double pole, they will ski at about 30 kilometers per hour. And you see when they use the double poling action, they cannot use it because it's too fast at the maximum level here, which then needs certain consideration when you use that particular technique. But for us, what we found was that the functional force and power velocity relationships in double poling of skiing is similar in shape as the force and power velocity relationships of an isolated muscle. This is not always correct. So for example, people have done studies on the speed of running as a function of the resistance provided to running. And that seems to give a very linear relationship. And similarly, people have measured power output in cycling as a function of pedaling rate. And again, there, the relationship seems to be much more linear than what we have found. However, when I look at those studies, they usually don't go very close to the isometric contraction, and they usually don't go very close to maximum speed of contraction. And maybe there would be also curvature there if people looked at a larger segment of the velocity, of the range of velocities than what we have done, because we almost went from maximum to minimum velocity in this particular study. So finally, I would like to very briefly talk about history-dependent properties of muscles. And in this segment, I would like to particularly talk about residual force enhancement, where we have done quite a bit of work. And so what do we mean by these residual force enhancement or history-dependent properties? What we mean by that is the following. Imagine you have a muscle here with an isometric contraction, so the length doesn't change, and you activate it, and you obtain a certain steady state force. If you now take that muscle at a shorter length, and you stretch it actively, then you get an increase in force that can be, to a certain degree, explained by the cross-bridge theory. Then you have a transient phase where the force decreases, and then it reaches a new steady state. And when you stretch the muscle even longer, you have the same, but your force at the end of the stretch is bigger, and your steady state force is higher as well. And this is what we call history-dependent properties. And this is not predicted by the cross-bridge theory. So here we are at the same muscle length, the same activation, all isometric, but we get very different amount of forces. And if you were to shorten the muscle, you would actually get less force. So you get a whole bunch of isometric maximal forces that depend very crucially on the action that has preceded this isometric contraction. And as I mentioned, this cannot be predicted with the cross-bridge theory. And that's why it has been very important for us and very interesting for us, because there is no accommodation for these history-dependent properties. And one of the things why I introduced the Titin filament shown here in blue again is because we felt very early on that maybe these history-dependent properties could be explained by the action of this third filamentous protein in sarcomeres, Titin. And very early on, about 20 years ago, we proposed the following hypotheses. We speculated that when you have a Titin here and you stretch it passively, so this is a passive muscle, no activation, then of course you get a certain amount of passive force, because we know that from a certain length on, we have a passive force of Titin that resists elongation of a muscle and the sarcomere. And we then speculated that when you have an active muscle, and an active muscle, of course, means that it's calcium activation, so you have released calcium from the sarcoplasmic reticulum, and the calcium activates your actin and myosin cross bridges. It might also activate Titin bind to titin and then make titin stiffer. So when you now elongate the sarcomere or the muscle, you get more force because titin is stiffer because of calcium binding. So we tested this particular theory with one of my postdocs in about 2006 and 7, where we took isolated myofibrils and we stretched them in low and high calcium concentrations. And we wanted to see what is the difference in force of titin. In order to only measure titin, we use myofibrils because the only passive force that you get in a myofibril is from titin, because everything else, all collagen fibrils, all other matrix proteins have been removed from this property. What has not been removed, of course, is actin and myosin interactions, but we then removed or inhibited actin-myosin interaction, in this particular case that I'm showing you, by removing troponin C from the actin filament. And as many of you will know, when you remove troponin C from the actin filament, then cross bridges cannot attach to actin, and you get zero force from actin and myosin. And then we can produce stretches at the different calcium concentrations in a myofibril and only measure the titin force and no other structural proteins and no actin myosin forces. And when we do that, then for the high calcium concentration, we get big amount of stretch force. And for the low calcium concentration, we get a low amount of force as we had expected. And this was done with many sarcomeres and myofilaments and myofibrils, and we then verified this result statistically. So the idea then was, well, if this really happens and the calcium binds here in this elastic element somewhere on titin, where would that be? And we focused on these areas here, the so-called immunoglobulin domains, primarily because there are so many of them. And we thought maybe if calcium bound to these immunoglobulin domains and made them stiffer, then maybe that could explain the result that we just have observed on the entire myofibril. So we recombinantly produced 8 immunoglobulin domains and then used fluorescence spectroscopy to see if calcium bound to the immunoglobulin domains, and then checked mechanically with atomic force microscopy if indeed we get a larger unbinding force and that we need more force to stretch these immunoglobulin domains. So initially we have here the fluorescence intensity for an immunoglobulin domain 27. And when we add calcium, then we see a depression of the spectrum. And if we add more calcium, then we see a further depression of the spectrum, indicating that calcium bound to the immunoglobulin domain. If we then take the calcium out, this goes back and is reversed, and you get the normal spectrum. So we then knew, we were fairly certain that calcium indeed binds to these immunoglobulin domains, and then we wanted to see if it made these immunoglobulin domains stiffer. So with atomic force microscopy, we unfolded these immunoglobulin domains that we had, and it takes a certain amount of force, with little calcium concentration as control, and then with an activating calcium concentration shown here in yellow. And then we did several hundred experiments in the control situation and in the calcium situation, and then found that indeed, in order to unfold an immunoglobulin domain in the presence of calcium, it took more force, about 20% more force, to unfold than to unfold an immunoglobulin domain in the control situation. So indeed, not only did calcium bind to these immunoglobulin domains, it also seemed to make these bonds stronger that need to be unfolded when you extend immunoglobulin domains. So we found, however, that the increase in force here, when we only did this, was relatively small and didn't really explain these force enhancement and history-dependent properties. In fact, we found that this would only explain maybe 10 or 20% of the entire force enhancement that we observed normally in muscles and myofibrils. So a second proposal we had is that maybe Titin might be binding to actin, which is relatively rigid, and therefore could not extend here in the proximal segment where it was bound, and it would only be able to extend here. And of course, when you make a spring of a given property shorter, then it becomes stiffer because the strain on this segment would then be much bigger than it would be on the entire segment. The reason why we thought this might be happening is because Titin naturally binds to actin back here anyway. And so we thought maybe upon activation, there is other attachment points that might then bind with actin and prevent elongation of this proximal segment. So we tested this by putting fluorescent markers on Titin using these fluorescently based antibodies that go to very specific places on the Titin segment. And the one that I'm going to show you goes to the PEVK segment, and then it looks like this. So here we have the antibody on the PEVK segment, and these light stripes that you see are the fluorescently labeled PEVK areas of a myofibril. And then we stretch this myofibril and we show some of the results here. So here in this brown dot, I'm showing the distance of this antibody that we placed on Titin's PEVK region from the Z line. So this distance will be plotted here. And here we do a passive stretching. And when we passively stretch, then we would expect this to elongate. So this length here to become longer. And indeed, if we did the experiment, we did about 125 experiments like that, then we indeed see that. And just once more, we stretch passively, and this seems to get longer. But the elongation is not as big as the entire sarcomere because this slope is bigger than that one. So we also know that this segment elongates as well. And now we do this with the activated muscle. So now we expected that this segment, the PEVK segment, would bind to actin and would therefore not elongate. So this would remain constant, which would mean the lengths here of this point would be constant and would be going parallel to the horizontal axis. But that did not happen. Typically what happened is that we got a little bit of elongation initially, and then there seemed to be some binding, or somehow this marker got stuck, and then the entire elongation was only in the proximal segment, and this segment did not elongate anymore, and we got the parallel line to the horizontal axis, this horizontal line like we expected. So once more, we have some elongation, and then a binding or a getting stuck. And once more, here both elements elongate, the proximal segment gets stuck, and the distal segment elongates. And so we think that this might be a part of the reason why we get this history-dependent increase in force after stretch that cannot be predicted with the cross-bridge theory. We also think that this might also play a role also on the eccentric part of the force-length relationship. So here we have the force-length relationship for concentric contraction, which I showed you. But in eccentric contractions, muscles become stronger than isometric. This flattens out somehow. And we think that this might in part be caused by Titin's stiffening, as I have shown. And also we think this because when we have an eccentric contraction, we need much less energy, much less ATP for an amount of force than we need in an isometric or in a concentric contraction for the same amount of force. So therefore, we think that Titin might play a big role here, not only in force enhancement, but in these eccentric contractions in general. And the reason why we thought that was based on an experiment that we now did almost 20 years ago. So we published this in the Journal of Experimental Biology. This was done on cat soleus muscle, where we looked at force enhancement at a long length of the muscle where we have naturally occurring passive force. So this is the passive force, and here we activated the muscle. This is active force. If we stretch the muscle passively from the short length to a long length, we get about the same amount of passive force. However, if we activate the muscle at the short length and then stretch it to the long length, we get the force enhancement that we expected. but when we then deactivate the muscle, we get an increase in passive force that we called passive force enhancement. And of course, initially people thought this was due to damage of the muscle and other things, and we eliminated all that and demonstrated that indeed the passive force is higher after an active stretch. And this then we showed also to be true in single myofibril experiments. Therefore, we could confidently say that this is likely due to Titin. We also found that when you make longer stretches, so here in brown we have a short stretch, in yellow a medium, and in green a longer stretch. When you go from a short to a medium to a long stretch, this passive force enhancement becomes bigger and bigger. And again, we verified that on the single myofibrillar level as well, again indicating that most likely Titin is responsible for this particular property, because in the myofibril, nothing else really could. So what we then proposed was the following little paradigm, where we say, well, this is the force-length relationship of an active muscle, actin and myosin, and we believe this is perfectly fine. So this is the plateau, this is the descending limb that is exactly proportional to the amount of actin-myosin filament overlap. But then what we thought is what happens to the passive force is that it doesn't stay stationary, but the passive force changes upon activation of a muscle, in the sense that the passive force occurs earlier because of the shortening, the substantial shortening of the Titin filament, and it becomes stiffer. So this force increases faster as a function of length change than here, because the Titin filament is stiffer. So because of Titin, we have an earlier onset of passive force and a steeper increase of passive force in an active muscle compared to a passive muscle. So if we look at this particular situation here at about 3.4 microns, then in the passive myofibril, we might have very, very little passive force, whereas in the active myofibril, Titin might contribute more force than the active isometric force that comes from the actin-myosin interaction. And we have shown that this is indeed the case on single sarcomere preparations where we tested this particular hypothesis. So in conclusion, and coming towards the end of my talk here, I wanted to emphasize that we think that Titin is a soft spring in passive muscle, and that's why you can move muscles very easily through the range of motion. It would not be good if Titin was stiff, or if you had spastic muscles that don't allow you to move really freely through the joint range of motion. But we think that Titin becomes stiff in an active muscle, and we think that this happens because of calcium binding to Titin that increases the molecular bond strength, and therefore individual Titin elements cannot elongate as readily, and also because that Titin might be binding partially to actin, therefore reducing its spring length and therefore become stiffer. So how might this possibly be interesting for sport applications? Well, there's very little known about what happens to Titin in exercise and disuse. For example, we don't know much, do Titin isoforms change with exercise or disuse? And do the number of Titin proteins change with exercise or disuse? We do know in rat models of disuse that the number of Titin proteins is reduced to about 50% from normal. So disuse seems to cause a decrease in the number of Titin proteins. And we also know that in children with cerebral palsy, we have done that study, that Titin is reduced to about 50% compared to normal in children with cerebral palsy. And we also found a relatively small but significant change in the Titin isoforms in children with cerebral palsy. So the question then also becomes, does Titin contribute to the increased force or work in stretch shortening cycles? As you know, in sport applications, we have a lot of stretch shortening cycles. And we published some work with my colleague Atsuki Fukutani, where we think that yes, residual force enhancement and Titin is very important in athletic movements that involve stretch shortening cycles and contributes to the increased work and power that we get in a shortening contraction that is followed by a stretch right preceding the shortening. So, coming to the end, what did I want to tell here to you today? The first thing that I wanted to talk about is the cross-bridge theory and the three filament theory. I wanted you to realize that there's not only actin and myosin, but that there's a third filamentous protein in sarcomeres that likely plays a crucial role in force regulation and contraction. I wanted to introduce the force-length relationship and explain that this force-length relationship, excuse me, is very much derivable from the cross-bridge theory. I talked about the force-velocity relationship in some applications and wanted to emphasize that this property comes out of the cross-bridge theory, but not naturally. It only comes out by appropriate choice of the rate constants of attachment and detachment of the cross bridges. And if you don't do that properly, then you will not be able to obtain a proper force-velocity relationship. And I also wanted to introduce some of the work that we have done on history-dependent properties. They're less well known, but in the past 10 years have received an enormous amount of attention, with more and more people looking at Titin properties and more people looking at history-dependent properties of skeletal muscle. So with this, I would like to thank Luis once more for inviting me to chat about muscles. I love to talk about muscles, so thank you for giving me the opportunity. And I'm happy to answer questions that you might have.
Well, thank you so much. Thank you so much, Walter, because your presentation, as usual, was amazing. And I think that I don't believe that some of the students, they were aware about how important Titin could be for the muscle mechanics. And I think that while students are warming up for questions, I have a question.
Sure.
And because when you were talking about the force velocity dependence, I remember about the fan effect. And I was thinking, how could we associate your studies about the third element within the muscle model and the fan effect?
Well, the fan effect, of course, says that when a muscle produces work, so when it's shortening, it can set free and requires much more energy. While before Fan did these experiments, everybody thought that when a muscle was activated, it had a certain amount of energy available, and that energy was constant, independent, if you had an eccentric or a concentric or an isometric contraction. And so I think the way the Fan effect or metabolic considerations might be accounted for is that we know in eccentric contractions, there is less energy required than in isometric or shortening contractions. And that can be very easily explained if we think that an elastic element like Titin becomes stiffer and binds to actin, because the binding to actin would only occur once and would require a very minimal amount of energy, whereas in an eccentric contraction, binding and unbinding of cross bridges occurs continuously and would require a lot of energy. And the other thing that we have observed is that when you are in the force enhanced state and you then shorten, you let the muscle shorten against no resistance, then the shortening speed will be greater than when you do that from a purely isometric contraction. So we think that this increased speed of unloaded shortening following an eccentric contraction is also due to Titin, and therefore provides energy to the shortening contraction that otherwise would not be there.
Okay, thank you. There was one question. How can, because you were talking about this, about trying to understand the muscle mechanics about this level, and then with this sliding theory, but how can we associate this with the other level of biomechanics modeling when we think about the whole system as a spring mass model?
Yeah, and that's a very good question. There are several people who have tried to model muscle from the molecular level up to the whole muscle level. And in fact, I'm working with a graduate student on that right now, and also with a colleague from the UK that tried to do that. And I help them in their effort, but I always tell them that that might actually be not a good idea. And the analogy that I usually give is when you think about a very simple engineering example. So for example, I have a uniform beam and I press on the beam and I want to measure the deflection of the beam. Then I can use a linear elastic beam deflection theory and I can solve the equation really easily, and it gives me a very, very, very accurate result. Or I could model the beam in all its atomic detail and all the atomic interactions, and then apply a force and then try to calculate what the beam deflection would be. First of all, the model would be infinitely more complex. And second of all, I guarantee you, it would give you a completely wrong answer. So I think one has to be very, very careful in trying to take molecular level events and scale them up to the myofibril level, to the fiber level, to the fascicle level, and then muscle level, and then to the in vivo human muscle level. There are so many other factors come in. So if I wanted to have a good muscle model, for biomechanical analysis, I would not start on the molecular level, but I would start maybe on the fiber level or even on a bigger level to kind of reconstruct that.
Okay. Daniela, don't you want to make your second question?
Yes, Professor. Would you think you can apply your study of the bikers, the bike riders? I like the idea of the position of the knee related to the pedal to the car race drivers.
Oh, I have I have no idea. I'm not familiar enough with.
No, but I think would be a great idea. You gave me some good ideas, so that's why I'm thinking.
Well, the thing is, you know, I think race car drivers essentially lay there in the car in a virtually reclined position, almost like laying in bed and being slightly propped up. And I don't know what exactly they need to do with their feet and what control of the force direction they need to have. But I would hope that car developers made the cars in such a way that they push that what they need to do can be exactly and easily accomplished.
No, of course, I think, of course, they can be easy accomplished. However, the performance, I'm talking about car drivers. What I see when you put me the pictures, because for the last 3 or 4 years, I'm following car drivers going into car races. And I think the best ones are the ones that also like to ride bikes. And I'm also trying to relate it with the position I see them in the car. And I also see a great level of coordination that you mentioned. Yeah. So that's why it's intuitively that I think maybe we can apply your study.
Yeah, so you're talking about normal car drivers, not car racers.
No, car racers. Car racers.
Oh, car racers as well. Yeah.
I follow a category that is named Mercedes-Benz Challenge. So they have car racers. They have some regular ones, unskilled ones like amateurs, but they also have professional ones.
Yeah.
And the ones that really race fast as close to Formula One, I think they are very well coordinated. So when I saw your bike studies in the beginning of the presentation, I like the direction you give to the study. So I'm thinking,
yeah, very interesting. I think the one thing to keep in mind for any sport or ergonomic type of an activity is, and that's why I like the bike example, because in the bike example, from a mechanical engineering point of view, it is clear that you would like to exert force always perpendicular to the crank, because that's the only force that produces work. But from a biomechanical point of view, it's not necessarily good, because we cannot exert force in all directions the same way. It's the same with my arms. If I push forwards, I'm much stronger than when I push upwards. Or if I have to push slightly backwards, I'm very weak. And very often I find that people do not consider that there's a biological system there that is not like a machine or a motor that just can produce a certain amount of force, but that force depends in which direction it has to be applied.
I have another explanation for your findings.
Okay.
My explanation is this, because I have two backgrounds, like Luis, that have several backgrounds and always
that is not
inspired.
Like a machine or a motor that just.
We have to look to when you are doing a research, the movement is very fast in many directions, and not everybody has a very coordinated eye. So I think when you did the first explanation, you are very correct. What is the problem with probably the guys that went on your research? For the lifelong, they never pay attention to that point that was essential. And so they develop their muscles to other conditions without seeing the essential. And when they try to be, because right now their infrastructure is, how can I say, not properly good for that, to perform that task, because they went a little bit into wrong direction. So I think you are right on your findings and your perceptions. However, not everybody is able to do. And during the research, suppose I am doing a movement, not paying well attention, I also need to input the time. Last week, one of the first lectures that Luis organized was about the time perception. So for a person that is learning for the first time, he also needs some time to adapt to the movement. And that's why you probably do not get the final performance. That is my thinking. I'm exchanging ideas with you.
Yeah, thank you. Yeah.
No, thank you for the presentation, because you are much more advanced. So I'm trying to catch and see. At least I have the ability to see who is better than me, and then I try to bring down and share my difficulties.
Yeah. Well, everybody has their specialities, you know.
Walter, and what about your thoughts concerning the Lambda model, about the idea that we shift the threshold of the stretch reflex?
Yeah, I probably cannot really comment on that very much. I have read about the Lambda model, but I'm really not a motor control person. Mm-hmm. And And I know that it is a very controversial model. Some swear by it and think it's the absolute correct idea. And I've had many discussions with Mark Latache and Anatoly Feldman about the lambda model, but I have never really done research to explore that. So I probably don't, well, I can't really give you an answer because I never really have done experiments myself to really explore this particular issue. I just know about the controversy. I know about the different positions in the field, but I have no research experience in it.
Yeah, no, I'm asking that because, for example, I see that according to your result, regarding your experiments, there are some mechanical reasons to explain what we can see about the force velocity behavior. Mm-hmm. And according to the lambda model, this is something that we can change because we can change the threshold. Voluntarily can change the threshold. Yep. Then my conflict thinking about this is how can we change the threshold and change the mechanics at the same time?
Yeah, well, sometimes I get invited to talk in motor control symposium, and my favorite topic usually is to talk about what you mentioned as the mechanical aspect. And that is, for example, when people do cycling activities or they do these, arm movements that are perturbed movements, I think very often what people forget when they look at muscle synergies, very often what they forget is that if you want to produce force in a given direction, or if you want to resist a certain perturbation that is occurring in a certain direction, then from a mechanical point of view, you don't really have an infinite number of possible solutions, but it comes down to very specific synergies that you have to invoke in order to respond properly. And how that is organized in terms of a motor control level in the brain and on the spinal cord, I don't know that. But from a mechanical point of view, I know that these synergies have to be there. Otherwise, you fail at the task. Yes. And sometimes I think when people look at that, they forget that certain synergies are just not possible when you have a certain movement or certain perturbations that you want to resist.
What about listen to your answer to Luis? I thought about your first slide when you put the, how can I say, when you say short mechanical options, because you put the shape of the muscles. And I think the problem is that not all the people have the perception about the different shapes of the muscles inside of you. Maybe on our cultural programming, we think there is only one way or two ways. And when you are challenged, you are not a researcher, you don't know the answer. So that gives like a black hole on your mental programming. Maybe it can be an explanation.
And I don't know. Yeah, no, I think that's an excellent point. That muscles have different structures, and therefore the different structure then also gives them different functions. And many years ago, when I was a postdoctoral trainee, so like 30 years ago, I did work where I measured muscle forces in the cat hind limb. And my favorite example is always show the force sharing between the medial gastrocnemius and the soleus muscle in that situation. Because when a cat stands, there are situations where it only activates the soleus muscle, but not the medial gastrocnemius. And when you put a little tape at the paw of the cat and he goes like that, it makes a paw shake response. The cat will only activate the medial gastrocnemius muscle and not the soleus. So here you have agonistic and synergistic muscles where you can activate one but not the other, or you activate the other and not the one. And depending on the intensity of the movement, the force sharing shifts from one muscle to another and is completely different. And that's what I love that particular finding that we had there 30 years ago, that you have these two muscles and they're always at the same place and they have the same anatomy and they're at the same place all the time. They don't change. But depending on what you do, they're completely differently used to a different extent, and they have a completely different contribution to the required movement, depending on what the movement is. And I think that exactly goes back to the structure of the muscle and the fiber type distribution and those type of things.
Walter, Ariane, don't you open your microphone and make your question?
Yeah, sure. Thanks for your presentation. And I'd like to know if does trigger point have any specific place to form muscles? In example, sarcomere, to reduce the sarcomere in size and consequently his contractile function, or something like this. Your opinion about trigger point?
I'm not quite sure if I understand the question. So you're talking about trigger point?
Yes.
Therapy and whether or not that can shorten a sarcomere?
Yes.
I'm not sure what you mean by shorten a sarcomere, because as sarcomeres, you know, they can have virtually any length. You know, they're made up of actin and myosin filaments. And let's say in a human skeletal muscle, the shortest sarcomeres, they can become as short as 1.2 microns, and then they can become as long as 4.2 microns. So you have an enormous range. You know, a sarcomere is not a given length. Depending on the muscle length, it will be shorter or longer. So when you say shorten the sarcomere length, I'm not quite sure what you mean by that.
I mean, not in a so specific way. Just because I read some paper about trigger points, some modifications that happen on sarcomere, but not with details. So that's why I've been thinking, where does it can be formed, or not sure, the localization of trigger points.
Yeah, of course, when you press on a muscle with a trigger point action, then of course you will press on a very specific part of the muscle, and that will affect thousands and thousands of sarcomeres, not only one. And the muscle in that region will be deformed. They change of the muscle or the sarcomere in that particular region. So I can see that, but I'm not sure what effect that would have.
Yes, thank you.
Thank you.
Walter, there is another question, but Alessandra, she has some problems with the microphone. Can you take a look on the chat?
Okay, there is...
Alessandra,
there's about 18 messages on the chat.
start with, in your opinion, what would be the first
Oh yeah,
one?
I see it. Yeah, I see it. In your opinion, what would be the contribution of the stretch shortening cycle in force production in the double poling in cross-country skiers? I think the contribution of the stretch shortening cycle in double poling would be quite big, because I'm not sure if you have seen cross-country skiers when they sprint at a very high speed, but they put their entire weight on the poles, and in the initial phase they stretch these muscles and then they pull through. And my imagination is that this pre-activation and initial short stretch would play a very big role in producing force and work during the shortening phase of the muscle. I don't think anybody has ever really looked at that very carefully, but that would be my guess.
And she made and there is another question. She made a second question.
Okay. Yeah, I see that. Considering that muscle force production lies in many aspects, molecular, muscle architecture, the neurocontrol, would you see the same trend in the force velocity curve in a participant with different backgrounds, e.g. physical inactivity or elite athlete. I think the force velocity properties would be affected by, by a sedentary person, an elite athlete, primarily because an elite athlete presumably would be stronger, and therefore along the force axis, the force velocity relationship would be shifted up. And then very often athletes have very distinct fiber type distribution. So for example, if the athlete is a sprint-like athlete and has a lot of fast twitch fibers, then that would affect the force velocity relationship as well. So then also with training, you can change the number of sarcomeres in series. And if you have more sarcomeres in series, that also gives you a higher velocity of muscle shortening that would affect the force velocity relationship. So my answer would be that yes, in detail, you will affect the force velocity relationship when you train and when you're an athlete. But generically, like in terms of the shape, would essentially the shape change dramatically? Then I would argue probably not. But the maximum speed, the maximum force, where exactly it stays relative to the action of your joint, that could potentially quite a bit.
May I make more or less follow-up question, perhaps?
Sure, go on, Thiago.
I'm just a little bit curious because during your presentation, you mentioned that ideally for such explosive movements like the double pole skill in cross-country skiers, athletes would like to maximize the power generated. But you did show that they are normally racing at speed that it's not really the peak power. Did I get this right, or what would be then the interpretation on how they are positioned in the power curve?
Yeah, no, you saw that correctly. According to our study, they would not really be working at an optimal frequency and an optimal speed. One way how this can be influenced to a certain degree is by changing the pole length that they're using. So in the olden days, people used longer poles, and that allowed them to have longer ground contact, and that slowed down the speed of the movement and the frequency of movement. However, in the mid-1980s, I think it was in 1985 or 1986, they limited that the pole could not be taller than the person is tall. And in classic skiing now, it actually got even more restricted. So now the poles can only be as long as 83% of the skier's height. And so therefore the pole length was artificially restricted, which makes it harder for the skiers to take full advantage of the poles. So if they were able to ski with longer poles, their power output would likely be greater, but they're not allowed to do that by the rules of skiing. And of course, we have many, many events where I think in most sports, we cannot really work at an optimal speed, let's say, or power output. When you think about a sprint runner, their contraction velocity is very, very high. And therefore, when they sprint at full speed, their force production is very low, but running at a constant speed requires actually very little additional work or power. And so there is other considerations there as well. So for a cross-country skier, that would mean if you know that normally you ski at about 30 kilometers per hour, then you should train at that particular speed to hopefully maximize your ability for power output that's very specific to your sport, even though it might not be optimal from an absolute point of view in power output. In cycling, that works a bit easier because you can adjust your seat height and you can adjust your gear to always pedal at a frequency that you feel comfortable at. And you might know that in cycling, some cyclists, they change their seat height depending if they go uphill or downhill on the flat. And my favorite cyclist from many, many years ago when I was a kid, Eddy Merckx, he was famous for that he put his seat height somewhat lower whenever he cycled up one of the mountain passes compared to when he was on the flat.
Okay, thank you. That was imagined in such a close, for instance, ballistic movements. If like a high jump or something like this, if this relationship would be somehow also shifted, because I can imagine you have a time constraint that you need to perform a high jump in order to be able to lift off the ground. So you cannot just maximize very isometric force or something like this, or otherwise you don't perform the movement.
Exactly.
And then I was thinking, then where do you train this? Do you shift the force velocity curve up? Do you try to increase the speed that you can produce? How do you go about finding the sweet spot for such movement? Since clearly it's not the force velocity, nor is it the power curve, but it seems to me, with very little experience, that it's somehow an external constraint. So how is it to find a sweet spot to work on athletes so that they can produce the best possible movement given the sports constraints?
Yeah, that's a very good question. And you know, the generic answer that I usually have there is, one thing we know is that muscles do adapt to what you train them with. So, you know, if you're a bicyclist or a speed skater and you work with very shortened hip muscles all the time because you lean forward to reduce air resistance, then those muscles will get used to work in a shortened position and they will adapt in terms of the sarcomere numbers in series and so on. And so we do know that muscles adapt to what you exercise them or train them at. And so the best recommendation, I think, is, you know, if you are a high jumper, practice high jumping. You know, if you're a runner, practice running. If you are a cyclist, and you know, that's why I always say in my lectures, in my muscle mechanics class, I always say there will never be a triathlete. There will never be a triathlete who will break the 10-kilometer world record in running, and there will never be a world-class runner that will break a record in cycling. And the reason for that is when you do triathlon and you practice swimming and running and cycling, you prepare your muscles for different things, and therefore you don't get the optimal running muscle, and you don't get the optimal swimming muscle, and you don't get the optimal cycling muscle. You get a hybrid, and that hybrid will never ever be as good as the person who only runs or only cycles or only swims, because the muscles do adapt to what you're doing.
Okay, thank you.
Thank you, Thiago. Sara?
Hello. Good afternoon.
Hello.
Thank you very much for the presentation, Professor. It was very brilliant for me. And I was wondering because I work with microgravity. The muscles and microgravity was a work I did during the college. So I know the masseter muscle, he do not affect by microgravity. And the other muscles have all the reductions of structure and size and reflex and everything, but the masseter muscle is not affected in almost anything. And a reason for that can be the structure of his fiber, because just have not much about it in the literature.
I was not aware of that, but that's a very interesting result. And I could see that maybe that particular muscle would not be affected because you still have, I think if I understood you correctly, it was the masseter muscle, is that correct?
Yeah.
Yes.
You still need that you still need that muscle for eating, presumably, and you might use it as much in low gravity as in high gravity, and therefore you might just really undergo the regular training that muscle has all the time, and that's why it might be less affected or not affected. It reminds me a little bit, you know, I live in Canada here, where we have, as you can see, still lots of snow and very cold, and we have animals outside that hibernate. And it's very similar with hibernating animals. You know, when you are sick and you have to lay in bed for 2 or 3 or 4 months, your muscles deteriorate greatly. But muscles of bears and other animals that hibernate, they are virtually unaffected. And so it appears that there is a possibility for muscles in certain way to be protected from atrophy, despite the fact that a bear might be sleeping for 4 months straight and not really move around at all. And their bones seem to be protected as well. And so I think there are ways of how some muscles might be protected from disuse, you know, like microgravity is kind of a disuse type of a thing. And maybe the masseter is one of these muscles, or maybe you use the masseter so often anyway, because you continue eating, that it's just not affected because of that. Has anybody ever looked at the masseter when you don't eat anymore? What happens in microgravity when you artificially feed the animal or you feed the person for 2 months and they don't use their muscles anymore?
Yes, it's more liquid. Food.
Exactly, yeah. You give them liquid
Yes.
food, you know, they wouldn't use it. Would that then mean the effect is gone and they would also be deteriorating and it's there for a training effect? Or is it something inherent in the muscle, like in a bear or in a hibernating animal, that protects the muscle from disuse atrophy?
Yes, I was thinking because it's enough just not use for eat process is enough for not be the same equal effect in other muscles. So it's interesting. But thank you very much.
I have a question for you. Actually, was this in an animal model or is this a human work?
That was human.
Human work. Oh yeah, very interesting.
Yes, thank you very much.
Thank you. Yeah.
Mm-hmm. Walter, do you think that your approach to understand muscle mechanics also can help us to understand the pathological states of muscle, for example, spasticity or tremor?
Yeah, I think so. You know, I've always been very much kind of a basic researcher and interested in fundamental questions, but we do some research in sport and we do some research also in rehabilitation. So probably the most research we have done is in children with cerebral palsy, because we are interested in muscular adaptations in muscles with cerebral palsy. And right now we have 2 small grants where we try to look at children with cerebral palsy and how we might be able to reduce spasticity in a different way than has typically been done. Because typically people have used Botox or phenol injections, but they are not very good. And we have shown that Botox injections affect the muscles really negatively in the long term with a lot of fat infiltration and fibrosis. And so we try to now directly use approaches where we can directly try to treat the fibrosis rather than the hyperactivation. And we've also looked on the cellular and myofibril level at the ultrastructure of muscles from children with cerebral palsy. And I mentioned it quickly, have, for example, found that children with cerebral palsy in their spastic muscles, the protein Titin is reduced to 50% of normal. So they only have about 3 Titins per half myosin instead of 6. So we are looking at these structural things and the functional implications.
Oh, that's cool. And do you know that could be similar behavior if we look at people, for example, stroke people?
Yeah, I think we have not looked at stroke, but stroke, they develop very similar spasticities, and we think that very similar mechanisms might be at work there as well. Yeah.
That's nice. That's nice. Well, students, do you have any other questions to Professor Herzog? Well, I don't think that we have any more. Well, Walter, thank you so much.
Pleasure.
Because
absolute pleasure.
Your presentation was amazing. I think that students, they could get a lot of the information that you wanted us to get. And thank you for your time. That time now is something that is now always was something very precious for all of us. And the time that you have spent with us was precious and was amazing.
Thank you very much, Luis. And if anybody has any further questions later on, or you were too shy to speak up in the group, feel absolutely free to send me an email and ask me per email. I'd be happy to try to answer your questions like that.
And Walter, I don't remember if I mentioned before, but students for the evaluation of this course, they will have to develop a research proposal based on the lectures.
Okay.
So maybe I'm gonna send you some ideas that the students has had.
Okay. Yeah, I'd be happy to look at that.
Yeah. Then maybe we'll see. Maybe some ideas we can try to turn that on reality and make some.
Okay.
And put students to make collaboration with you.
That would be very nice. I know that a lot of people work in applied fields, and one of the areas that I think I'd love to work with people is if people potentially were able to obtain muscle biopsies from a variety of different populations, as you were mentioning, spastic muscles from stroke, or also athletic populations, or populations that are sedentary due to disease or so, to really look at more the ultrastructural level on the sarcomere level. That would be something that I'd be very interested in, for example.
Yes. Oh, I can give you another example here, because here we have Adriani. She works with the Army.
Yep.
She runs the biomechanical lab in the Army. In Rio. So
hello.
Hello.
So Adri, that is an open opportunity for you, actually, and yes. you have all these soldiers under your command.
More or less, because we didn't have a lot of biomechanical instruments. We actually have only 2 force platforms right now. Yep. But I'm working with Luciano Menegal at UFRJ. He has a complete biomechanical lab, and maybe
very nice
opportunity. Thank you.
Good. Nice to meet you.
Nice to meet you too.
Okay, thank you, Walter. Hope to see you soon, and not in a remote mode, but
yeah, I hope to see you soon. Always love to come to Brazil. Been so many times to Brazil.
Yes. Well, best regards to all of you, and see you. Thank you.
Thank you very much. Have a good afternoon. Bye-bye.
Bye-bye. You too. So, students, thank you. Oh, Walter, before leaving.
Yeah.
I put in the gallery view, and then I'm gonna ask students to open their cameras. Then you can, and ask them to say hello to you.
Oh, okay, good. Oh yeah, lots of students. Fabulous. Hello, everybody.
Yeah, that's nice.
Good to see everybody.
Yes, yes, that's something that is also good when you can see the faces.
Yeah, yeah. Very
Okay.
nice.
Okay. Thank you, Walter.
Thanks a lot. Have a good afternoon. Bye-bye.
Bye-bye. So now we're gonna end the streaming. Full screen.